everything that I have. Has everyone been able to see their grades for things in Moodle when you go in there? You can see. Yeah, so that's a good way to keep track. If there's a missing assignment, you know, it's not too late. Complete it, bring it to class, get some points for it. Likewise, continue to hang on to the tests, quizzes for future. There'll be one more quiz and then final exam in this class. And as far as this week goes, after we reviewed the test from last week, we'll begin photosynthesis. Photosynthesis, there's potentially a lifetime there, and I want to keep it very narrow to how it connects to the things that we've already talked about. And then there is a homework assignment associated with that, and the supplemental reading. There's not much in the Botany for Gardeners book on photosynthesis and respiration, so I pulled this from a university publication. It's fairly thorough, and so again, I want you to connect especially with the lecture material there. And all of this is to be done to say, if we get through the material, then the homework will be due Wednesday. If not, we'll complete it Wednesday, and the homework will be due on Monday. So just keeping up with those things as we go. And I'm going to pop this on and talk about it. Overall, I think the results were very good. I could see development in people's answers from the quizzes into the test, and some clear improvement, and especially on some questions where I know I talked about just getting that little core, that little nugget out of there. So for the true false section and the multiple choice, I marked the correct answer so you know what it is. Roland, do you want to make up the test now? We're talking about the answers, right?
Oh, then I'll leave.
Yeah. Leave but not take it?
Yeah.
So yeah, try to be back like at quarter to 2. But there's a couple I wanted to call out in there that were commonly turned up. And the first one was number 4, carrier proteins are used in both active and passive transport. And I just wanted to remind you, got all my presentations set here, about this one. Where I've outlined the 3. And so, right, this is the active, and this is passive. So not entirely, right? Diffusion osmosis doesn't require, but these two both. That was 3 true and false. Here. Yeah, absolutely, of course, as we go, all of them are can be discussed. Most monocots are wind pollinated. Yeah, that just came from the discussion in the dispersal methods about like the grains, grasses, and the structure of those pollens. And number 12 was the next one I wanted to, no, 13, sorry. Main groups of plant tissues. Right, the correct answer here was C, dermal, ground, vascular, and meristematic. And come back to this one now. Vascular we know a lot about now is that lymph flow, right? Epidermal we kept in mind as the outside. We talked about root hairs, right? We talked about leaf, waxy cuticle, and epidermis, the location of those guard cells, controlling stomata in the ground tissue. We talked about calling it cortex in the root. That's where water is moving past to get into the vascular system. We talked about it being pith in stems, and then being the parenchyma cells, palisade, mesophyll, or spongy mesophyll in the leaf. And of course, the one not mentioned here, most importantly, is that meristematic tissue. That's right. That could then be any one of these three down the line. That was one I was very satisfied I said that. We understood meristematic tissue and what it means, why it matters. Other questions? Number 15, the answer was F, about the things app nodes may contain. Right? I think most people correctly understood axillary buds, meristematic tissue, leaf primordia, and it seemed like adventitious roots was the one that tripped people up. So here, I'm not saying that they always do that, right? But it's really a question about, is that possible, right? May contain. And because of the other, right, meristematic tissue listed, that is definitely within the possibilities. We talked about that adventitious rooting with the strawberry plant vegetative propagation as an example. And in 19, small guard cells open because of B, an increase in turgor pressure. We spent a bunch of time looking at that. That seemed to be that D, both A and B, was the one that tripped people up. And A, not enough water in the cells creates the opposite situation, right? So without that water, there's no turgor, right? And then stomata remain closed. And with sufficient water, they swell and open. And that comes from our leaf material. Sufficient water here, and then you can see the vacuole, right? They kind of make it skinnier there to indicate not enough water. So those are two opposite things. A and B.
But a consequence of them being closed would be excess CO2.
The consequence of them being closed would not be excess CO2 in the leaf. If they're closed, then the small quantity of CO2 is going to be consumed.
Oh, okay.
Does that make sense?
Yeah.
Yes. 16, low transport dissolved organic minerals and carbohydrates. 16.
Sorry, what was it?
The answer is C, flammable/all direction. That could be.
I'll mention that later.
Yes, so all direction there is correct. I get that, then that's either whatever. Get in the zone sometimes. All right, so that was all for those. The rest of the short answers. I definitely want to mention how these. First, number 12. We saw before in the homework about annual rings, right? We looked at them a bunch of times. And here, we mentioned that situation where because of consistent light, right, in the tropics, we're seeing less change, right? There's no clear, because the seasonality is relatively consistent in terms of the growth potential because of day length. Right? There's not that huge hard stop like we have here where now, right, no new xylem is being made, and then next season we're going to see expect to see from the model. Yeah, keep the stems. Not open the stems yet. We look at one of these guys. Right? When the growth is like slow and constant, we see it would be something like this, right? Where it's hard to interpret where one ring starts and finishes versus a clear distinction. And again, so that's just trying to get at the environmental conditions impact on how the plant responds. And I had several people respond that it was because the tropics is wetter, that was the result, that the xylem tube changed size in response to that. And because you were getting at the right point, that there's something about the conditions there, right? I accept it, but I want to be clear that we don't know. There is dry tropical forest, right, where there's a wet period and then a prolonged dry period, just like, you know, our climate here. And then there's consistently wet, and of course elevation has an impact on that too. So it's not, well, you know, tropics is not exclusively wet in the same way. So, but they are beholden to their position in terms of latitude, and the light is a known quantity. We can look at any given time of year and say there's this much light. And generally, within the bounds of 23.5 degrees north and south of the equator, it's between, you know, 11 and 13 hours. So quite consistent compared to our, you know, 7.5 to 15. Right? It's either some number twice it, or it's a difference of 10% versus 100%. So that's 10 times different at some point in the year. Differences between the upper and lower parts of the leaf. What does the plant get from organizing the structure in this way? Right, this is just getting you to understand how the structures within something connects to the function. Right? We looked at a bunch of different views of the leaf. We saw stomata generally located on the lower side for water conservation. Big cubicle, again, for water holding on the top, and big palisade musical for photosynthesis up there. So that's what I was just getting at, is how is this thing laid out and what are the functions? Walking over there reminds me, Akasha is kindly recording these classes and then posting them to Moodle. So feel free to take advantage and watch them for a little bit of review, see what somebody asked about a question. And of course, if you miss a class, chance to see it too. Yeah.
Even like the first class?
I don't know if she has all of them. I think it's been the last couple weeks.
Yeah, I didn't, it was, at least after the second week, I don't have the first. Yeah, it was around the second, third week when I asked. Okay, so,
Thank you very much. That's awesome.
yeah.
All right. So, number 14, where in the root branch roots form, and why is it essential that they start here? I'm gonna give you a clue right now. That one's going to be an extra credit question on the final exam, and then you'll know it. I said number 14 is going to be an extra credit question on the final exam, and you'll know it. So, the first part, what I really wanted to get across here is that the purpose of roots, right? When you think about roots, if you had to sum it all up in one word, what would it be? What's that? Roots? Water absorption. Yeah, right? And so here, it's essential that they're connected to that vascular tissue, right? And so they come out of that layer of meristematic tissue right at that section. So I gave an example of this back in roots. And this was you imagine here now, right, this cross section is a vertical root, maybe it's a taproot or something larger. And this is going to be a lateral root spreading that capacity for water absorption out into the soil. Has to be connected here, right, to xylem phloem, or immediately, one, it's not being fed, and two, its whole purpose, right, is to move that water into the mainstream, right, of xylem in the plant, and right through to the flow, but the root, we got that water coming up, and we don't know quite yet what's coming down until it's established. Yeah.
So the pericycle is another term for vascular tissue?
Pericycle or endodermis is like this one circle around the outside of it.
Around the vascular bundle?
In this case, it's around the, just inside the Casparian strip surrounding the whole bundle, yes. And so that's meristematic tissue where that can be produced.
Quick question.
Yeah.
Is the pericycle itself meristematic tissue or just in contact with it?
It's, when you get in there, it's a little bit like, if you're looking at it under a microscope, that cambium is meristematic. And then the pericycle is, it's like one cell layer over as the ring around it, but it's directly connected to that. Yeah. So it comes from that cambium meristem tissue, and then that's like the piece that is wrapping the whole bundle, but now you're inside it, so it's like these two tubes are connected instead of just slightly separate. I promised plants have a problem and what can we do about it. This one, right, was the opposite example from the homework question, right, where stomata were open all the time. Now, right, these guard cells don't have vacuoles, so they're slumped shut, closed all the time. What do you think this would do? And how does it compare in the case of the cactus? So, just in a general sense, good or bad for the tree? Bad. Shut means, to there's an earlier question, no CO2 in, that's not good. No water out, right? That's going to be problematic if we can't open those stomata. So overall, net negative. Would this be more damaging in the cactus? Why or why not? I was looking for you to point out one of a few different things here. The overall answer is less damaging. One, because what's one good reason?
No water loss.
Less water loss at least, right? Because right, the stem of the cactus is water storage, and its leaves are much reduced, right? Those spines have way less surface area to lose water, and thus, by having less surface area, fewer stomata, right? So definitely, not as bad. And some people pointed out correctly too, which we'll get to either today or Wednesday when we talk about photosynthesis in cacti, that they only open, this is the cactus now, only opens the stomata at night, which is another way that it would be not as bad, but that's an additional bonus fact that we have to share. Yeah. And 16 was another, something else has gone wrong. This plant doesn't have palisade mesophyll. What are you going to do here? And I said give two ways the plant could adjust this limitation, right? Some of the, hold on one second. The plant could adjust, right? So I was looking for answers that indicated you understood what was lost without the palisade mesophyll and a potential way to make up for it within. And so some said, okay, that means this thing is not going to be doing a lot of photosynthesis, which is true, so put it in a low light area. Right? Well, that's not something the adjustment the plant can make. Or, give it more water, for example. So these are all, right, condition improvements that we would be making from the outside. And I wanted to see that connection with internally what's possible. So the first place to go with that is, what do the palisade mesophyll do? Which is what? They do photosynthesis in those chloroplasts, right? So here you can see, right, this is more chloroplasts and some, but not as many in that spongy. So we're losing some photosynthetic capacity. So an easy one would be start using the spongy, right? To make more chloroplasts there. Some even describe that likely with that gap, the whole arrangement would shift. Probably these things move up, they fill in more space, make more of those, right? So expand the capacity of like this kind of plan B backup absorption level. Is it a good one? What else could be done there? In the same way that some of the answers that were like, oh, just basically put this plant in the best conditions for a plant that can't do a lot of photosynthesis, if you work backwards on the plant side, there's some similar things that could be achieved, like closing the stomata, right, to reduce water loss, basically slowing down growth. It's going to be slowing down growth means less overall need, which means less sunlight might be okay. Some indicated changing, the balance of shade and sun leaves, making more sun leaves to do more absorption, right, from a place that's going to be in direct contact because of that missing capacity here.
I wrote it kind of different now that I look at it, where I wrote it could grow more slowly, so it wouldn't need to photosynthesize as much, but it would actually realistically be more like it would just grow more slowly because it could photosynthesize as much.
True, but yes, I mean, in the short term, that's going to be the result.
Anyway, is that it would adapt by growing more slowly since it could just utilize what photosynthesis it had available from the sun.
That's right. I mean, there's still, you can only use the resources available, but it's also going to try to get more, right? It doesn't just, it's not just satisfied with that change, right? It's still trying to absorb and survive. And so, what can it do, right? I mean, in reality, when that happens, right, when there is damage or when there's a loss of function somewhere, then the other network does step in and do more. Or if for whatever reason in a greenhouse, say the light source changes, then the arrangement changes, or maybe the leaf moves, or which leaves are doing more work changes. So there are those responses too. The next one was about Parthenocarpy. That was from our fruits discussion last week. And lots remembered banana example. Keep in mind here that what's key is that a fruit is produced without pollination or fertilization, and that connects to many mentioned no viable seeds, and that's right. But just saying that it's a vegetative propagation method is not so specific. Taking a cutting is also a vegetative propagation method, right? But there's no fruit involved, right? So this is specifically a fruit production without those traditional pollination and fertilization steps. Seed dormancy is the next one. It was well answered. I think it was right. It was fresh in our minds. We just talked about it last week. You know that the root comes out first to get water, and everybody correctly said that the purpose of dormancy is to favorable for survival at germination. And then got one more puzzle. Right? You find this setup of a leaf, and you're supposed to tell me something about the condition. A lot of palisade mesophyll. Right? So I went from none in the couple questions ago. Now there's a lot. Big cuticle, lots of stomata, and I'm saying what's the light, temperature, and humidity right here, and what makes you think that. So. And relative humidity. So one thing we know from this, densely packed mesophyll, lots of stomata, and cuticle. I was trying to connect it here to something from leaves that I showed you and that you drew in the homework. Sun and shade. Here. So, dense mesophyll, right, a double layer of it even, for example, indicating high sun. So we know that light here is high. That thick cuticle, lots of stomata. People correctly intuited that those are water retention strategies indicating that temperature is high. And in terms of humidity, there was really no wrong answer as long as you explain. I think it's a toss-up on do a lot of stomata mean it's humid or mean not. It could go either way. You could have them open or closed, right? But that you understand that it's the manipulating of those that's going to respond to the conditions is what matters. So this one was variable, and we know that it's a high light environment. It's probably a high temperature environment, which again, when we get to talking more about cacti and their photosynthesis, we'll see the interesting change that goes from high temperature in the daytime to low temperature at night because of all that heat loss to a clear sky every night in the desert environment. So again, you're seeing in these short answer questions, I'm just digging in on the basic concepts and asking you to explain and see if you understand. So here in 20, I was going back to roots again, and we had talked about the two primary types of root systems. So if you imagine this is the tree, this is the drip line of that tree, which is, we think, maybe that's the canopy, right? And we're saying this whole space in between, no grass. And that's because those fibrous rooted grasses do that good job of absorbing right at the surface and easily compete with the young tree early on for those water resources, right? Go all the way back to roots. So that's something like this. So if that's right up against your young tree, then unless there's deep watering, which would be the way to respond to this if you didn't remove the grass, right? But if you're just depending on precipitation here, for example, where it's generally smaller increments of lighter rain, then potentially most of that's getting sucked up by the grasses, and the tree is not maximizing its growth potential because of not enough water. So you had to mention something about roots here. Talk about the competition for water. Cross-pollination, again, was fresh in our minds and well described. We spent time last week talking about it in the homework, looking at stuff like this. You had to show me at least that you knew cross-pollination was two different plants within the same species, okay, and mentioned something about that there's this journey getting stuck onto the stigma, right? Dividing and becoming the two, falling two, and then eventually reaching ovule fertilizing the ovary in the process. So, as we were reviewing last week, I was talking about those stories, and that was story number one, the story of pollination, and the other that I strongly hinted at was story two, the journey of water from the outside in. So we looked at this in the presentation, we looked at this in the homework, getting water from soil environment into the vascular system, and now it's being extended in the xylem up, right, through those vascular bundles, and then out via stomata as guard cells open. So there was a couple extra pieces from where we started in roots as we added in what we know about leaves. And of course, it began with this sort of story, and one of the words in there to use was vacuole. It may have been here where you talked about vacuoles manipulating the concentration gradient to get water. It might have been guard cells becoming turgid, right, to open up and let water out. At least one of those mentions. And then of course, right, spent a lot of time talking about that Casparian strip and how we keep that line of water always, that chain connected, so we avoid cavitation, that air bubble inside. This was drawing a 3-year-old tree. This was just like in the homework. And a woody dicot angiosperm, even in bold for you. Excuse me. And to get full points here, you had to identify that angiosperm tree, right? Means that we have a combination of larger vessels and smaller tracheids. Otherwise, we got most of the points for that. Twig ID was straight off the sheet, and likewise flower ID came from here. And to remind you about this part of number 25, circle the terms that describe the flower below. Right? We had male and female parts there in the drawing together, which would make it a perfect flower. And of course, then, all perfect flowers are also monoecious. Indicate that you understood both pieces there, certainly both. There was a chance for some extra credit on the 8, 9, 10 labels there. Sepal, the receptacle, and the peduncle connecting that flower to the plant. And number 26 was the bonus. The primary words of plants increase in size. This is, they make new cells through cell division, and then those cells get longer. Now we're gonna, and elongate, right? So cell division and elongation, two major ways that cells grow. So cells increase in size, which leads to plant. If you said something about, plants get water and nutrients and they get bigger, right, you got a couple of points. But I was looking more specifically at, in terms of the actual physical change, it's you either make more or you make what you got bigger. joint, if you will, from here's the feed-up ovary connected back to the main plant. And there's like this joint below the ovary that's almost like the stand for it. And so, right, receptacle, sequel above, and then feed-up below. And both of those. Other questions? All right, well then let's take 5 right now. We'll come back to the
That was it, and I'm like, okay, that's good. Do you need this still? This is there. Like, looking back, that's right now. Like, come up with these answers. No, that's how I feel. I'll be like, how did this like?
If I just look I know, I feel like that's the only thing you can say. It's just like, you're not just saying it, but it's not.
So yeah, I'm looking at the notes, obviously, yeah,
Yeah, like combined.
there is, yeah. Is it punticle?
All just funny names.
Oh, you did? Oh, yeah, I'm really excited about it. So you're working like underwater then?
Yeah, I hope.
Oh, yeah, she should be like the one who's like big one, like her, the two assistant like her, kind of split the farm. So it should be helping. Yeah, we're like the three rangers, three greenhouses there. That's just come back. Really excited about that.
What are you doing now though?
Actually, I came for
It's usually a problem.
yeah, exactly.
Yeah, I mean, you have to have IBM kind of to be
I definitely
in the grower.
I was there.
I feel like going to see it. Well, warehouse on my birthday.
I just think it's good that it's
Otherwise, you don't know what you're telling someone else.
how long you can keep the story going.
Big weekend there. Friday,
Also,
it's a while to get this stuff. Yeah, it's what you gotta do. Do whatever you can.
did you want to go?
I did not go. I did much time. I got too much into it. Wasting time. But you're just doing one of those.
Yes, I think I'm doing that whole thing. He's got a partner, but I don't
How was the harvest for cash?
Always a good idea. I have some big toenail about to fall off. That drives what do you call it? You blow up the tires? Alright, let's get back in. So first, an announcement from Sport Club about our holiday party. The date has been selected and everyone is invited, family, etc. Thursday, December 11th, from 6 to 7:30 in 117, 118. Anybody been to one before? Holiday party?
Yes, I do.
So if you haven't, it's cool. It's kind of a send off. You know, maybe some people are graduating. Other people, it's just like there's 3 weeks until you're taking another class here, and there's food. There's door prizes. There's little gifts for kids and a dessert table to share. So the Horta Club funds the meal, and then everybody brings dessert to share. And we hear from different groups. This time around, there's going to be a slideshow and thank you to Loretta, our administrative assistant, who's retired at the end of December. So chance to say goodbye there too. you're thinking about coming, please sign up. And of course, if you change your mind or, life happens, don't worry about it. But we just like to get an idea of how many to get the space right and get the food order done.
I'm shooting myself in the foot saying this, but they actually do give out pretty good prizes. And like, for
Hey,
the
bro, come
lottery.
on, bro, come on.
You did your chances are not as good now. Other people.
Somebody told me last year, so I got it.
Yeah, there's like, there's hamburgers and tools and, some plants and wreaths and, yeah, it's nice.
Bro, you're not missing out on anything.
Two birds on your shoulder. Anyway, if you come, it's a nice event, and my coworkers in the department will be there. Your cohort of students, lots of people will be there, and if not, no problem. But it's a nice thing to put on my own club to kind of send off into the holiday season where maybe we don't see each other for a while. So now. Speaking of going down into the holiday season, let's talk about light, photosynthesis in particular. And so when you hear roots, you think water. When you hear leaves, you're thinking specific things. Here when you think photosynthesis, you're thinking light, right? Photo is in there, photosynthesis. Put those things together, making things, synthesis, building up things, and photo light. Making things with light. And so here's a couple ways to look at the basic thing is carbon dioxide and water combined with light absorbed in the chlorophyll of chloroplasts in plants gives you glucose and oxygen. Okay, so this is the essential equation. It's at the end of this presentation also. And you'll notice that respiration is virtually the flipped equation that we get there. And in terms of the chemistry, right, our equation's balanced, so there's this many carbon dioxides and all that, but don't worry about that, right? But do know what goes in and what comes out, right? That's essential and connects directly to what we've just been doing, powering germination of that seed. Right? Talking about how a leaf looks and functions related to what sort of light conditions it's in. So keep that in mind. Okay? CO2 and water. Here there's light. We understand this to be energy. That's abbreviated like that. Energy gives you glucose. Here, say sugar and oxygen. So interestingly, the plants using CO2 and releasing oxygen, right? Which is, we're actually having CO2 and inhaling oxygen. It's convenient for us. Right? And of course, this comes about when stomata are open, carbon dioxide in, water comes when roots are successful at taking up water, right? Through that whole process that we discussed. Light comes when leaves are successful at absorbing sunlight into the mesophyll. And then put those things together into these sugars, right? Glucose is like the base unit, and then you get a bunch of those stacked up and you can make glycogen, which is the starch for storage, but we'll just talk about it as sugar energy. Okay, and so now, there's more words on the page, there's the name of some chemicals and stuff, and you don't need to know all of them, but it's there. Some are specifically interested in more context, and sometimes, so I want to have that available, but also I'm going to outline specifically what you need to know here. So just in general terms, right, there's two sets of reactions in this process. The quote, light reactions and the light independent reactions. So understand that, right? It happens in two parts. One of them does not require light. When I was a kid, they called it the dark reactions, and now it's a rebrand to to the light-independent reactions because it can happen without light. It's not that it only happens in the dark or somehow depends on darkness. And where this all happens is in the chloroplast, right? So this is a single chloroplast model. The light comes in, absorbed in some place, water comes in, right? And this stuff happens. So we get sugar, we get a little bit of energy in this ATP package. This happens in the chloroplasts, and like I said, respiration does the opposite. So keep it real simple so far. There's two parts. One is the light reaction, and the other light independent, also known as the Calvin cycle. So, understand how plants maximize this situation. It's worth taking a little look at the qualities of light. So, light's made up of several wavelengths. Shorter wavelengths have higher frequency, that means more waves, and thus more energy packed into them. And when we're talking about this, we're talking mostly about this narrow rainbow strip of the visible light spectrum within the whole electromagnetic spectrum, right? From radio waves and infrared waves, then you get red, right? And then violet, and then ultraviolet. So from highest energy on the right to lowest energy on the left, out here you're talking about, right, space things that might cause you cancer over time. In the visible light spectrum, we're talking about a range that means red is the lowest energy light, and purple, right, blue violet, is the highest energy light. So understand that part. And of course, green happens to be somewhere in the middle, which is that color that's reflected for what we see. So here again, a bunch of names of things. Keep it real close to the ground. Light is absorbed by these specific pigments inside the chloroplast. And we call them chlorophyll A and B. Chlorophyll A and B work according to this graph, right? A is the red line, B is the green, and you can see they both have two peaks. They peak in the blue-violet range, and they peak in the red-orange range. And so, this is the peak is where the most absorption is from that chlorophyll. Right? So chlorophyll A is really good at getting that orange-red light and that far, almost ultraviolet light. Chlorophyll B, really good at getting that blue light, and kind of does, get some orange. What none of them do is get that green light, right? Because that's reflected back, and thus the color that we see, the chloroplast, the plant leaf, the stem, etc., as green. So it's primarily, right, blue and red that are giving most of that energy. And then some of the other colors are also absorbed by a secondary set of pigments. And this is, just for knowledge's sake, carotenoids, sounds like carrots. They reflect orange and absorb others. And you can see the blue line there. Anthocyanins reflect blue. That's like what makes a blueberry say look that color. Xanthophylls reflect yellow. That's all to say that these pigments work in different wavelengths to absorb as much light as possible. And the color that's reflected is the color that we see. So just for the sake of it here, chlorophyll, right, absorbs light energy. And it does it very effectively in the blue-violet range and in the red-orange range. Finished.
Filter the light spectrum information. Are we saying that no plants absorb out of the visible light spectrum?
Oh no, they definitely do.
Oh, okay.
But the primary energy is coming here. Like the beyond ultraviolet, it's the intensity is such that it could be damaging, and below infrared, there's not enough to push a lot of growth. So it's like this Goldilocks zone, sweet spot of appropriate level of energy that's neither too little or too much. Yeah, Julie?
Is that kind of an explanation, or part of an explanation of why chlorophyll and those plants are green? Because it's kind of that medium where they can get a lot of spectrum? Sorry.
Yes, that's right. So, I get back to the one more down the road here. At some point, as those pigments were evolving, they were able to extract the most benefit at these 2Ps and didn't do a good job of getting the light in here. But it was okay because they were effective in these other areas. So that isn't to say that, right, there could be the evolution of a new pigment that's maybe really good at absorbing green light at the expense of the others. But for now, because things, kind of the ball got rolling in that direction, now everything else depends on plant signaling. A lot of it is in response to the quantity of red light sensed or the quantity of blue light sensed. And so that's why, like, if you're doing, you know, most things now are LEDs that are full spectrum, but once upon a time there was like high pressure sodium bulbs that gave you more blue-violet light and, more red-orange light, and metal halide bulbs that gave you more blue-violet light, and you would try to use both of those to simulate the scope of sunlight over the course of the day. Yeah, Roland.
So chlorophyll A and chlorophyll B are obviously central to the photosynthesis. Am I right in understanding that like carotenoids and anthocyanins is more about like protection from stuff, or does it also play a role in photosynthesis as well? I know that it like protects against sun scald and things like that, but
yeah. So carotenoids have some photosynthetic role in terms of energy absorption, but you know, it's maybe like 5% of the overall thing, whereas these two are like 94%. And then anthocyanins, anthophils, etc., it's more about those polyphenolics that you're talking about, that like, oh, this is going to be for plant defense, right? Or this is going to make it smell or taste a certain way or something like that.
Are the carotenoids located in the mesophyll cells, or are they? I mean, they're
This is
obviously
all inside the chloroplast,
In the
inside
chloroplast.
the mesophyll cell. Yeah. So yeah, once upon a time, chloroplasts were their own little, you know, basically blue-green algae, their own little organisms that they could turn light into food. It's pretty cool. And then they got, this was just like out in the environment. Likewise, mitochondria were their own little organisms that could make energy, and then early plants were like, hold on, if we put these all together, then we can both make food, extract energy from that in the same place, and we can grow a lot faster. So these early single-cell organisms doing simple function were collected together to make more complex organisms. So this one is showing you pretty much the same thing as the last. Just that the peaks are in the blue, and again, here the lower number means more energy, strangely, and here in the red. So take that into consideration for both this discussion of photosynthesis and as you're growing plants in general. I was talking about the different lighting effects. It used to be you need to figure out if you're going to do supplemental lighting, how you're going to get blue light, how you're going to get red light. Otherwise, you're not going to maximize growth, and you're going to get certain effect by acting like only one part of the spectrum. So let me put that maybe while I talk about this. And now we've mostly full spectrums. But both of those are distinct from these fluorescent lights, for example, that give off like a little bit, right? It's kind of in the middle, there's not much light intensity, not a lot of energy there. You know, it's a little bit on the red side. And so when you're using supplemental light, right, you're recreating the outside environment, the impact of absorbing sunlight at certain wavelengths, and it changes throughout the day. So for example, does anyone know when it tends to be more red light, for example, versus blue?
No.
Sunset, sunset, sunrise.
Sunset, sunrise, like that? Yeah. Early in the day, late in the day, that's when we see this red light, right? Lower energy light. And middle of the day, as it's heating up, right? The angle of the sun's getting higher, we're getting more of the high energy blue spectrum light. And so this is going to connect later when we talk about hormone signaling. This is how the plant actually senses days or nights, is by the amount of red light absorbed over a period of time. And so, back to my original point, if you're only providing, say, that high pressure sodium light that is heavy on the orange-red spectrum, then you're mimicking that sunrise sunset level that may not push enough energy for successful fruiting or flowering of the crop, or it might slow down growth. And then when you want to encourage that flowering, right, you want to provide that high value, low frequency, high frequency, low wavelength light, so that that flowering plant that's consuming a lot of energy now has more available. So you can change that input over the development period based on the need in indoor condition control growth.
Well, you had full spectrum lights though, right?
Mm-hmm.
How would you go about that?
Well, then it's just about time, really.
Leave them alone.
See what?
Leave them alone.
Yeah, exactly.
Well,
Oh, I know what you're talking about. I was just trying to make sure we understand that.
And some of them, if there's variable control, you can adjust, like, hey, you know, more output midday and less output, and you're like fully mimicking.
Yeah, I'm
Right.
trying to bite.
But at the same time, you're trying to grow as fast as possible, so then it's usually just like, okay, you know, I'm gonna give 16 hours of daylight here at the full spectrum level versus other plant, or I wanna keep this plant vegetative, I'm just gonna put it at, you know, below 12 hours. You're kind of choosing there then about time if all the light source energy is equal. Speaking of light, so we've got two sets of reactions here in photosynthesis. So we've got the whole thing, which I'll often abbreviate like that, photosynthesis, just to save a little time. Again, this one's energy. This letter E, that's photosynthesis. We have photosynthesis, and it happens in the light reaction and the Calvin cycle, aka like independent reaction. So here, you're just in the light reaction, still inside the chloroplast. Get some details about where it happens, but just know the chloroplast, right? It's essential, I think, to get that part. So there's a couple different photosystems. That's just about how the energy is moved along. The important part to get, I think, is that the role of the light does is, say, it excites the electrons in the chlorophyll, right? So basically, the energy packet in a photon of light, right, imbues more energy into like the solution that is the chlorophyll, and it's absorbed and heightened energy. So now it's it's almost like electric, like it's absorbed this high energy sunlight, and you can do something with it. So it's like this, the chlorophyll. Is this kind of solution where it can absorb the light and gives it more energy, right? And now we can work with that additional energy to push the process forward. So again, detail, don't get caught up here. So far I've talked about light absorbed, right, into, this is all the chloroplast here. This energy, extra energy in the chlorophyll. That the first thing that energy does is tear apart a water molecule and release that oxygen. So again, it's not essential to know every step in this process, but why I mentioned this is to connect with the importance of water, right? Of appropriate irrigation of anything that you're growing. Right? As soon as you're in a water deficit, you're immediately decreasing photosynthesis. So earlier I wrote, right, carbon dioxide plus water, light. Any one of those things is in short supply. Now the process is no longer maximized and you're not growing as well. So for optimal growth, we want to keep sufficient water supply, connects to that whole story about roots, so that the next things can take place. So it absorbs that water, excuse me, absorbs that solar energy, it splits that water, which is two hydrogens and an oxygen, so it breaks that up. And then oxygen is released. It does that step again. Oxygen is actually released as O2, and two of them join together. And so, right, this energy now continues to move through this electron transport chain that makes some ATP. So that ATP, when we're talking about respiration and photosynthesis, ATP is our energy source. That's just the name of the chemical structure that allows the plant to get energy, right? This is another kind of secondary energy carrier, but all these things are holding on to energized electrons that came from the sunlight, absorbed solar energy in the chlorophyll, and now we can move that, that absorbed energy around. So the first step, light reaction, is absorb energy and split water. So again, right away, don't have enough water, the process is stopping in the first step. You're already slowed down. Essential to meet that water need. The transport chain, there's videos associated with that if you want to dig in on the electrons moving around. But again, that's not the focus here. I want you to think about just the raw ingredients and what comes as a result.
Is photosynthesis one?
You're right, they're out of order. Is that what you're getting at?
I don't think that's my only point of confusion.
Okay.
Is one of them light dependent, and the other photosystem light?
No, this whole thing is in the light dependent portion of the reaction. And that was just, they were discovered in reverse order, even though the process, they figured out the process actually went this way, but this thing was identified first and they never changed.
But both of those are required for light dependent. Neither are part of light.
That's right. Yeah. So far, we're still under the light reaction. And now, to the light independent reaction, aka Calvin cycle. So again, I talked about not in the dark. We're still in the chloroplast, right? And the important piece in making this transition. So again, the result of photosynthesis is glucose, right, and oxygen, but these sugars that we can rip apart to make energy. And the result of respiration is the actual energy that then does the growing. But in this light reaction, we get a little bit of energy, which again, came first from the sun, and then we were able to transfer it down this line of, in the chloroplast membrane, and extract a little bit of energy from the light reaction to power the next step. Like, right, one ball had to roll downhill and knock over the domino to get the next thing going. So split water and then make ATP energy that powers the Calvin cycle. So if you think about the cycle like a wheel, right, that little bit of energy from the light reaction has to push the wheel start turning or start rolling. So that's one that is important, is that a little bit of energy comes out of this part. Here's the results here. A little bit of energy and split water to get split water into oxygen. Right? So here we've got the oxygen out already and enough energy to get the next step going. So the next step, the light independent reaction now, is where we make carbon compound, which again is glucose. The final product, glucose, is C6H12O6. And so you're going to need carbons, hydrogens, and oxygens to get there. The next important point is the presence of rubisco. This is an enzyme. Enzymes are biological catalysts, things that speed up a reaction. And this one is able to grab onto carbon dioxide molecules. And so this catalyst in the Calvin cycle, which was powered by a little bit of energy from the light reaction, breaks apart the carbon dioxide into these just carbon molecules. Okay, so the goal is to try to build this thing, so we're going to need carbons, right? And we're going to need hydrogens and oxygens that are coming from the water. But to get the carbon part, we got to rip apart CO2. So we're on the Calvin cycle side. Now we split CO2, and we start building them up. So all these things, G3P, it takes 3 turns to create one of these. Don't worry about that. The point is, all this stuff happens, right? Literally, that little bit of energy pushes the wheel around this enzyme rubisco and grabs onto carbon dioxide. And as this series of events happens, right, the result is turning a CO2 into just a C and then sticking it with the next turn onto another C and another C. And it just happens that the biologically efficient way to do it is to make a 3-carbon molecule and then later stick those 2 things together, 2 of those to get 6 carbons. But again, it's not a chemistry class, and I'm trying to stick to the important foundation of just the creation of the glucose and where things come from. Okay, so in the Calvin cycle, again, a light independent reaction, we split CO2 using rubisco, this enzyme that grabs carbon. And that's going to come in to be especially important later, how we grab onto carbon S, right, the chloroplast, trying to effectively make some food.
Quick question. Is the Calvin cycle produced from any other source besides ATP, or is it just the ATP that fuels it and then it makes the storage thing?
Yes. So it's, you need the initial step flight reaction happens, that then the ATP comes out of that like flips the switch to the next step, that then now that ball can roll downhill and start happening.
It said stored energy before, so I was wondering if there was something other than ATP that was the input in the first bullet.
Well, there is, yeah, so here, an ATPH also, go back here. So, again, this one, it's another
Okay.
energy carrier.
It's like a short-term energy.
It's just a way to like, you know, it's sort of the way that the fertilizer has an inert stuff and then you stick the actual nutrient on there. It's like a chelator that is a neutral thing that you can plug the energy unit onto, move it to the next place, and then pull it back up. And so I don't want to get too lost there, and so that's why it's just like ATP. If you know ATP is energy, I think for this, that's so to your point, those reactions work together. Right? The light energy, again, in makes the first step happen. Right? That's where water is used, oxygen is released, and it powers the next one that grabs onto carbon. Grabs onto carbon dioxide and makes it into sugar. So, again, if this one makes sense, without, like, you have to know all the little molecules and stuff of the process, and the key is that sunlight is filled with energy, right? Without that, this whole thing wouldn't matter. But it's coming at us all the time, and chlorophyll inside the chloroplast has the capacity to absorb a bunch of this energy and make this process happen. It's unique. However, RUBP, or Rubisco, this important enzyme that is our carbon dioxide bonding agent, has a weakness. That weakness is that it's not very selective. It will grab carbon dioxide when it's present, right, and tear it apart and get that carbon, and then the process rolls, puts those carbons together to make the glucose, to make the sugar that's going to become energy. However, it will also bond oxygen, and when it does that, rather than sugar that's going to become stored energy as the output ends up being carbon dioxide, which we need to go into the process to get energy, right? So that's problematic. And that is called photorespiration, when Rubisco bonds oxygen instead of CO2.
So it ends up like sending it out the other way.
Say again?
So it ends up like sending it out the other way.
Yeah, so it sends it out the other way, but the main point is you don't get the food, right? You don't get the food that becomes energy.
It creates CO2 instead of the.
That's right.
So you're kind of wasting.
You're wasting, absolutely. And in fact, you can see there's even a little bit of an input of energy to extract that CO2 in photorespiration, right? So that all around, it's cost you energy. You haven't got more energy out, right? And so you really want to avoid this situation. Really.
This is what scientists have been playing around with fixing, right, to make photosynthesis more efficient? Like this is like have you heard about this one?
I mean, a little bit. There's only so,
I think it was Rubisco was the issue.
you,
Which makes sense.
on a big scale, well, so plants have responded. Plants have a couple of cool responses here. But in terms of what humans could do, we could put in some other more efficient enzyme, but then we'd have to figure out a way that that would be in the replicable chromosome and be created the next time, right? So that's the challenge. So to my question here, so obviously this is bad, right? So we want to avoid this, and we want to get this, right? The process happens, and the plant gets fed, right? The plant gets energy. When do you think photorespiration might be happening? Andrew?
When there's too much O2 around.
Yes, and when, or the corollary, when there's not enough carbon dioxide. with respiration, we're gonna get there. So that one's in the breaking apart sugar to get energy, and when that one happens, you get lactic acid gives you a little bit of energy, whereas aerobic respiration gives you 16 times more energy than that. Anyways, you go cramping, right? But so yes, it's definitely in that territory. So when stomata are closed, CO2 can be limited. Okay? So this is the big risk. Spent that time in leaves talking about the opening and closing of stomata and the essential role of keeping enough water, right? Water retention, all those strategies, leaf hairs, sunken stomata, right? However, that all has to be balanced against the need for carbon dioxide to do photosynthesis. So when it comes down to it, right, those stomata are going to have to open to get CO2 if there's not enough, and that's going to cost water. So again, enough water is essential to keep photosynthesis going, right, and of course, to keep the rest of the plant processes going too. It's tricky though, because air is only about 420 ppm CO2, right? I mean, this is rising with industrial pollution, basically, but it's still a tiny percentage, right? 0.042% of the atmosphere. In contrast, oxygen is almost 21%, nitrogen is 70%. So plants are basing their whole thing off a very small part of what's in the atmosphere. So they need something that has a really high affinity for carbon dioxide. And that makes it stressful, right, and likely that photorespiration will happen when it's very hot and or dry. And of course, if you put those two things together, right, that hot and dry, then you're really amping it up, because stomata want to stay closed to save water for two reasons. But they need to open to get enough carbon dioxide to create food, right, in photosynthesis. So this slows down the plant's ability to grow, right, reducing how much energy is produced. And in the case of photorespiration, CO2 concentration drops, and the process with oxygen instead continues, which we just showed, takes a little bit of energy and makes CO2 instead of making food. And the only response, right, the only way to get around it is to open stomata and get more in. Because the atmosphere, right, it's always available. And of course, in controlled growing situation, if you really want to maximize that growth, you can also add CO2 to the environment, right, at a higher percentage to speed that growth, reduce that need, right, to open and close a lot, make sure it's sufficient, and get the most maximized growth situation. But plants didn't have that option, and they came up with some really cool solutions. They came up with two other photosynthetic pathways. The one I've been talking about is called C3 photosynthesis. Everything I've said so far is in the C3 realm, and that's because it's called C3 as in 3 carbons. If we go back a bit, Rubisco makes these 3 carbon molecules. And Rubisco also has that willingness to bond oxygen, right, and not make food in photosynthesis. It's a little tricky. So 3C3. The other two are C4 photosynthesis and CAM, or crassylation acid metabolism photosynthesis. And plants have developed these to survive in those very hot, very dry environments that I just mentioned are so risky for C3 plants because they force them to open stomata all the time. Okay? So about 90% of plant species are C3, and the remaining 10% are in these two. This is a sugarcane field. It's a C4 plant. Okay? In this case, high temperature, not necessarily dry, oftentimes humid tropical but very hot. And so it can do C4 photosynthesis and maximize growth in the face of high temperature where a C3 plant won't be able to excel in the same way. And here's how they do it. So we're back in the leaf now. And here's kind of the traditional setup, right? So you can imagine this is a sun leaf, a couple layers of palisade mesophyll, right? Spongy, stomata, etc., and a vascular bundle dispersed throughout. In the case of the C4 plant, now, there are fewer vascular bundles, and they're surrounded by that mesophyll, right? By that capacity to actually do the photosynthesis in the chloroplast, fully surrounded and separated from that airspace. Because when stomata are open, it's the same as the outside atmosphere, right? It's clear passage. And so here, right, water could be lost from that direct connection. So here, we're both preventing some water loss and making sure that the energy is immediately available right on the outside. So C4 plants have figured out in space how to do it more efficiently, right, to reduce their water loss and thus keep stomata closed more than C3 plants can do. And you do that by stomata are always partially closed or partially open, depending on how you think about it. But point being, just slightly open all the time. So less energy flexing, open and closed, less water lost from being fully open, always a slow supply of CO2 available to flow in. Then, to Roland's point, they've employed a different enzyme. It's called PEP as an abbreviation that doesn't bind oxygen. And so it only grabs carbons. So there's no photorespiration. Photorespiration, as I mentioned, that over here, this one, but it can cut down efficiency by 20 to 25% if you get in that loop where there's no carbon dioxide. So that's a big drop. So it creates these 4 carbon compounds first, okay, and then it uses that Rubisco, okay, in the bundle sheath cells that are not in direct contact with the air to make those sugars. So examples, I talked about sugarcane already, corn, wheat, oats, crabgrass, a lot of monocot grass species there, some important agricultural crops, and many of which came site of origin from high sun areas, and in the case of corn and wheat and oats, also dry areas. So responding to those 2 pressures, they came up with this strategy again. Get a different thing for pathogen to bond carbon dioxide to get the carbons and separate in space. Where the process happens. That's this one, right? So, don't worry about that. Just kind of want to look at how the whole process connects. But I wanted to show this, especially in terms of efficiency of the use of available carbon dioxide and the difference that it makes to do C4 photosynthesis versus C3. So here's the CO2 concentration on the x-axis, and on the y, how much can be taken up. So dark line is C4 plant, right? So even at low concentrations, right, it's already maximizing here, where C3 hasn't, it's almost halfway to its max efficiency. So, quite a big difference. All this area is then capacity for growth that C4 plant has that C3 plant can't get. Not to mention, there's a whole gap here where it can't even start the average C3 plant until enough CO2 is present because of that effect of the different enzyme, right? The Rubisco grabbing it versus the PEP grabbing it. So clearly more efficient. Question about that one so far?
They use Rubisco and PEP.
That's right.
They just kind of coordinate it up from each other.
Yep.
So the open air where there would be a potential where C4 then Rubisco could bond to oxygen. They just use the PEP at that stage instead of Rubisco.
Yep.
But they still do use Rubisco, it's just behind closed doors.
That's right. So they've added a step. Should be more efficient. Yeah. So, right, carbon dioxide's coming in through the stomata. Photosynthesis is happening here in the chloroplasts of the mesophyll. That PEP, right, is grabbing all that carbon at this site, and then it's moving into the bundle sheath where it's now not connected anymore. So as it moves, there's no loss, and then Rubisco is doing the job of putting the carbons together. So by separating that out in space, it's reducing the loss of CO2, thus making it more efficient.
That's where the glucose wanted to end up anyways, is right by the vascular tissues.
Exactly, yeah, because the next step then is either one, store it somewhere, or put it into the phloem where it's going to be stored somewhere or used immediately. Yep.
In the C4 plant, the other cells that are not the ones forming a ring labeled as mesophyll cells, are those also mesophyll?
Like
one,
these, for
yeah.
example? Yes.
Okay.
So it'd be like the palisade mesophyll and the spongy mesophyll.
Okay.
And that gets us to, all right, so CO2 use in C4 plants, more efficient than the C3 plant, and to photosynthesis. It's named after plant family Cassalaceae that where this was first identified. It's the most efficient water use strategy. There's relatively few. There's probably, I said 10% of plants use one of these two. This is maybe like 3% of plants species. Most efficient water use strategy. I hinted at it earlier. The stomata are closed during the day when it's the hottest, right, in direct sunlight, and open at night when temperature in the desert areas has dropped significantly, right? So just by lowering the temperature, what happens?
Relative humidity increases.
Exactly. So less water loss, right? So we can take up that carbon dioxide at night, right, and store it for use during the day. Because, back to the beginning, there's a light reaction that requires light-dependent reaction. So it has to happen during the day. And so it uses this stored overnight carbon source to be employed during that light reaction rather than having to keep stomata open during the day to get the CO2 in real time to feed at the moment. So it uses this storage, right? So it grabs it with paths just like C4 plants, and then it stores it as what we call malate, which gets converted back to CO2. Again, just get the concept that it stores carbon dioxide from nighttime to use during the day. The impact of all this, right, only stomata open at night, having to store it, and then, right, changing that back and forth in forms, all of that takes energy, which means growth is slow. And that's why, right, this cactus could be 150 years old. Right? Growth is slow, but it's effective in the succulents use this method. Cacti we've been talking about as a model. Pineapple sort of does sit out as a good example of lots of goofy things like the aggregate fruit. So, I didn't leave space on this side of the board for it, but I can. In terms of water use efficiency, right, this is the most efficient. Then C4, and then C3. So here, got PEP, Stomata, open at night. Here, there, Stomata, always partially open. So open and close, always a little bit open, and then only at night. Very good water use efficiency, but slow growth. So it helps to, compare these things. Both of these alternative strategies, if you will, alternative pathways, both use PEP to fix carbon dioxide and to grab it and avoid photorespiration. The big advantage over C3 photosynthesis, none of that oxygen screwing up the process, reducing the output, reducing the growth. They both employ the Calvin cycle, and C4 uses the strategy of separating in space, right, where those things happen. And CAM uses separating in time, so that day-night difference, opening at night, storing that carbon to be used during the day, whereas C4 is still working during the day in the height of hot, potentially dry weather, but doing it in a way that reduces water loss and maintains a constant supply of carbon dioxide. Overall, photosynthesis increases as the light intensity increases, but it plateaus at a certain point, where it also increases as temperature goes up, but it decreases at a specific high temp. So eventually you hit some threshold where you can't grow any faster. So you either need to add carbon dioxide to the environment, so it's talking about it, controlled growth, right? You can do that. Or you need to, or the temperature needs to be raised to increase the rate of photosynthesis further. So as you change one, you can change the other to get more growth to a point. And eventually there's a point where temperature is too high, proteins are going to denature, right, and things start breaking down. So no more growth at that point, or vastly reduced growth. So this is to say that we can use our knowledge of these things to keep plant growth in the beneficial zone for effective photosynthesis by understanding the water need, the carbon dioxide need, and the corollary effect of stomatal opening or closing based on conditions, as well as light, right, which starts this whole process. So here I'm just talking again about how environmental factors impact the growth. So how much CO2 we have, how much photosynthesis we get. Right, so here carbon dioxide is a limiting factor. As we get more of it, we can do more photosynthesis, and then now we've topped out. We can only use our CO2 so efficiently, and now it's either it needs to be warmer, we need more water, or all conditions are as good as they get, and this is the plateau. In terms of temperature, each plant's going to be different, but there's an optimal temperature for photosynthesis and a range. Right? So in this example, this species could actually photosynthesize below freezing, right, and up to around 104 degrees. Now, you're getting very little growth at that level, but not killing the plant. And so, just to show, right, here's how they respond. And of course, if you look at generic generic C3 plant versus generic C4 or CAN plant, this whole thing can be shifted to the right as it can handle higher temperature and still put on growth, albeit slowly. But some slow growth here, right, at really high temperature is more than nothing, right? And it might be equal to what the average C3 plant could do, say, in the 90s. Most of our C3 plants, food crops, for example, have an optimal temperature around 80. It's fairly, generically accurate. And then as you get beyond 90, things really start slowing down. These plants are working on staying alive, conserving water, right, and making sure that they can feed the existing structure they've already created rather than making a new fruit or a new leaf or expanding further, which is the same thing they're doing on the other end when there's either not enough light or temperature is too cold. And so it's not just a constant increase forever. There's this zone that's most effective, and by noting that, we can work with it. If the temperature is a little bit higher, but the water need is still met, right, then we can, and the carbon dioxide availability is present, then we can continue, right, to grow. But if temperature rises and we're not irrigating more, then that's going to affect that growth capacity. So all the factors, right, combine to determine how effective, how quickly we can develop that individual plant. Okay, so today we talked about photosynthesis, and in the next we'll talk about respiration. Keep this in mind for then, when you've got the two pieces to compare. So for today, some takeaways from the left side of the column. Photosynthesis happens in the chloroplasts. It uses solar energy from the sun to start. It gets carbon dioxide. It's a building process, right? We're creating different molecules that are going to be broken down later in respiration to get food. It needs water. Oxygen is released. It produces these sugars, and it only occurs, right, needs that initial light for the whole rest of the story that I just described. So I got through there. That's the last one. So that means the assignment is due on Wednesday. We'll go over that and try to make sense of photosynthesis as we move into respiration, which kind of, as you look in the other side of the mirror, I think it helps understand which one is forming it together.
Oh, yeah. Oh, so he has all of the supplies ready to go for next week. So you can give your little talk and you can actually just do that on screen. I had no idea he was doing that.
I didn't realize that. So you said I know, wherever I have to go.
So Spread the word, and now you're done. Yeah.